This video is about membrane transport, the process by which substances move across the cell membrane. Substances are transported for chemical reactions, electrical potentials at the membrane, and for elimination from the cells. The cell membrane is a complex structure surrounding the entire cell and cell organelles. It is fluid in nature, allowing molecule movement, and consists of a phospholipid bilayer and proteins. Proteins like ankyrin and BAN3, along with cytoskeletal proteins like spectrin, are vital for membrane structure and function. The cell membrane has a lipid bilayer with hydrophilic heads and hydrophobic tails, organized in a fluid mosaic model. Various proteins like integral and extrinsic ones, as well as glycolipids and cholesterol, are embedded within it. Functions of the cell membrane include acting as a selective semi-permeable barrier, maintaining different compositions of intracellular and extracellular fluids, and receiving both chemical and electrical signals. Transport across the cell membrane can be passive or active. Passive transport includes simple diffusion, facilitated diffusion, filtration, osmosis, and dialysis. Active transport includes primary and secondary active transport, and endo/exocytosis. Passive transport occurs through the lipid bilayer or protein channels, which can be leaky or gated, such as voltage or ligand-gated channels. Simple diffusion is the movement of molecules from an area of higher concentration to lower concentration until equilibrium is reached. Diffusion can occur through the lipid bilayer for lipid-soluble substances like oxygen and carbon dioxide, and to a limited extent, water and urea. Electrolytes are generally impermeable. Substances can also diffuse through protein channels, which can be open or leaky, like Na+ and K+ channels, or gated, opening only under specific conditions, like voltage or ligand-gated channels. Factors affecting the rate of diffusion include lipid solubility, molecular size and weight, temperature, membrane thickness, surface area, and concentration, pressure, and electrical gradients. Fick's law of diffusion describes the net rate of diffusion as proportional to the concentration gradient, permeability, and surface area, and inversely proportional to the molecular weight and thickness. Facilitated diffusion is a passive, carrier-mediated transport for larger water-soluble molecules along the concentration gradient, involving a receptor site on one side of the carrier. In facilitated diffusion, the rate of transport is initially proportional to the concentration gradient, but reaches a maximum velocity or Vmax due to the limited number of carriers. This slide appears to be related to the previous one, showing a graphical representation or a conceptual illustration of the transport rate reaching Vmax. Carrier-mediated transport has peculiarities like specificity, competitive or non-competitive inhibition, saturation, and blocking of receptors. Examples include the transport of glucose and amino acids. Osmosis occurs when two solutions of different concentrations are separated by a semi-permeable membrane. Water molecules diffuse from the lower to the higher concentration solution. Osmotic pressure is the minimum pressure needed to prevent osmosis. It depends on the total number of dissolved solute particles, expressed as osmoles or mosmoles. Isotonic solutions have the same osmolarity as plasma, around 290 mmosmoles. Cells in hypotonic solutions swell, while those in hypertonic solutions shrink. Gibbs-Donnan equilibrium explains the concentration difference of diffusible ions separated by a semi-permeable membrane when one compartment has non-diffusible ions, like proteins. This slide illustrates the change in ion concentrations as the system moves towards Gibbs-Donnan equilibrium, influenced by concentration and electrical gradients. Two conditions for Gibbs-Donnan equilibrium are: all solutions must be electrically neutral, and the product of diffusible cation and anion concentrations must be equal in both compartments. Diffusion potential, or equilibrium potential, is the potential generated across the cell membrane due to non-diffusible ions. Its magnitude is calculated using the Nernst equation. The Nernst equation calculates the equilibrium potential for a specific ion. The Goldman-Hodgkins equation considers multiple ions and their permeabilities. Filtration is the movement of fluid and solutes through a membrane due to a pressure difference. An example is capillary filtration, determined by net filtration pressure. Dialysis is the separation of larger dissolved particles from smaller ones. It is used to eliminate waste products from the blood in cases of renal failure. Active transport is categorized into primary active transport, secondary active transport, and endocytosis, which includes pinocytosis, phagocytosis, and exocytosis. active transport is carrier-mediated, rapid, and moves substances against the electrochemical gradient. It requires energy expenditure by transport proteins with ATP-ACE activity. Carrier proteins in active transport demonstrate specificity, saturation, competitive inhibition, and blocking. Substances transported include Na+, K+, H+, Cl-I, glucose, and amino acids. Primary active transport examples include the Na plus K plus pump, Ca plus plus pump, and H plus K plus pump. Energy from ATP hydrolysis drives a conformational change in the carrier protein to transport ions. The Na plus K plus pump is an electrogenic pump. It binds 3 Na plus inside and 2 K plus outside, activates ATP-AS, and pumps 3 Na plus out and 2 K plus in, maintaining cell volume and high K plus concentration inside. The Na plus K plus pump is a major energy-using process. Its regulators include cellular Na plus concentration, thyroid hormones, aldosterone, dopamine, insulin, and inhibitors like ubiane or digitalis. The Ca++ pump is present in the membrane of the endoplasmic reticulum, mitochondria, and cell membrane. It involves a uniport carrier and helps maintain a low Ca++ concentration in the intracellular fluid. Secondary active transport depends on the Na plus concentration gradient created by primary active transport. The carrier lacks ATP-ACE activity. The substance is transported along with Na+. Examples of secondary active transport include co-transport mechanisms for glucose and amino acids reabsorption, countertransport for H+ secretion, and Na-KATP-AC indirectly affecting Ca transport in the heart. This illustration shows glucose reabsorption. The Na plus K plus pump maintains the Na plus gradient. Na plus and glucose are co-transported on the luminal side, and glucose exits via facilitated diffusion on the basal side. Transporters can be classified as uniport, synport, or antiport based on the number and direction of molecules they transport across the membrane. Extracellular material is brought into the cell by endocytosis, which includes pinocytosis, receptor-mediated endocytosis, and phagocytosis. This process requires ATP, Ca, and microfilaments. Pinocytosis is a type of endocytosis where the cell membrane invaginates, helped by proteins like dynamin, to form an endocytic vesicle, bringing extracellular fluid into the cell. Receptor-mediated endocytosis is a highly selective process for importing specific large molecules, requiring energy and calcium. It involves coated pits and proteins like clathrin, actin, and myosin. Examples of receptor-mediated endocytosis include the uptake of low-density lipoproteins and viruses, such as hepatitis and AIDS viruses, as well as excess iron. Phagocytosis is the internalization of large particles, bacteria, or dead tissues by specialized professional phagocytes, like certain white blood cells. The material contacts the cell membrane, which then invaginates. This illustration shows the process of phagocytosis, including the formation of pseudopodia, internalization, fusion to form a phagosome, digestion, absorption, and the resulting residual body. Passive transport requires no energy expenditure, follows the concentration gradient, and may or may not require a carrier. Active transport requires ATP, can go against the gradient, and always requires a carrier.